We began this process about 3 years ago, recognizing that cancer really is a genetic mutation-based disease, generally acquired, but also inherited genetic mutations, and that pharmaceutical companies were beginning to develop drugs targeting the products of those specific mutations. And as a result, in order to identify the right patients, then you need to be able to perform diagnostic testing to identify those mutations in individual patients. So that's really the fundamental basis of why we took this approach and incorporated the technology of next-generation sequencing and array CGH to detect not only point mutations but structural abnormalities in individual patient chromosomes as well within their tumor cells. And we initially partnered with a large clinical trials oncology group to develop a panel of the specific genes that needed to be tested that would elucidate the genetic mutations related to today's FDA-approved drugs as well as those that are in clinical trials. We introduced that panel a year ago into the clinical research environment and have subsequently partnered with Genospace, a bioinformatics company located in Cambridge, Massachusetts, as well as Thomson Reuters, in order to develop a commercially available product to offer to the general population. We're focusing on intractable or recurrent disease of all types of cancers to help answer the question, once you fail standard therapy, what's next? What can you do next? So Genospace provides the big data platform and the algorithms that help determine the clinical relevance of individual genetic variants. Thomson Reuters provides a global knowledge base of genomic medicine and the world's literature that's kept up to date in a curated database format. And we apply the specific information on an individual patient to the global knowledge database so that we can then deliver an individualized or personalized and highly specific, precise report of information to an individual oncologist or the patient about the specific characteristics of the disease, what literature supports the clinical relevance of the findings, as well as what clinical trials are available to this patient based upon their individual characteristics.